Hello, I'm here today to present to you our senior design project. It's our capstone project for the mechanical engineering department here at the University of Alaska Fairbanks. We were tasked to design, print, and test an environmental DNA sampling device in collaboration with the fisheries department. The project was mentored by the lovely lady on the left here, that is Dr. Jessica Glass. She has a lot of supporting research that was provided through us by her graduate student, Maris Goodwin, and her project is funded by the state of Alaska and the Natural Science Foundation. In the center of the photo there, we have Andrew Wilson and myself on the right-hand side, George Deal, and together we've developed the team Fishy Filters to move through this project. So many folks may not be familiar with environmental DNA and how it's used. So essentially, it's anything that's shed off of an organism into an environment, and that information can be collected and applied to research to do things such as track these lovely invasive species that we have here. On the left, is a northern pike, one of the primary invasive species here in Alaska. And on the right is a European green crab, which we're seeing more and more of in some of our coastal regions. This design can be used to track and monitor some of these the presence of some of these species and be able to move forward and take action and how to limit that infiltration. Currently, environmental DNA has been collected either through active sampling or passive sampling. The design on the left uses a vacuum pump to draw fresh water through a environmental DNA filter. But this method has its limitations as it can only take a brief snapshot of the environment, whereas the passive sampling method on the right is able to be left for a more prolonged period of time and collect data for sets up to 24 hours. This design also had its flaws, which left Jessica coming to us for a better design that works for the research that she's performing. This is the design that we were left with at the end of that time period. This is a model that's 3D printable, is affordable, and can be attached to a buoy line in marine applications so that it can be left for a broad period of time and collect a larger window up to 24 hours of the organisms that would be present in a biome. We built a testing enclosure to be able to see how this actually functions and performs in a simulated current, and our testing provided that it was able to orient properly and move the way it should, as well as collect the sediment samples that were required. And where do we go from here? Well, we presented this idea to the American Fisheries Society about a month and a half ago, and there were many interested parties who wanted to incorporate this into their research. Thank you for your time and have a great day.